Hello and welcome everyone to this podcast for genetics. In this podcast, we're going to talk about our first two types of chromosomal mutations. We're going to first talk about duplications, and then we're going to talk about deletions. Both duplications and deletions share some similar characteristics, so it's appropriate to talk about them together here. As we talk about each of these, we're going to describe how they look at the chromosomal level. We're going to talk about how they form. Then we'll make sure we talk about some meiotic considerations. What do the chromosomes have to do during meiosis if they have duplicated or deleted regions? And then we'll want to talk about how they can change a phenotype. But first, let's talk about how they appear at the chromosomal level. So let's draw a chromosome here, and let's draw some genes on it. And we'll be fairly generic here. We'll just call them by single letters. Okay, A through H. And we'll put a centromere right here. So let's first talk about what happens if duplication occurs. And when this happens, one part of the chromosome will be copied. So let's say now we have A, B, C, D, E, F, G, and H. And you can see here what's happened is that the chromosome has been duplicated right here, E and F. So it's been copied twice. When they're right next to each other, like we see here, we call this a tandem duplication. If they're not right next to each other, either the EF has been placed far down on the other end of the chromosome or maybe even on a different chromosome, we would call that a displaced duplication. But what we see here is a tandem duplication. Now, while we have it here, let's talk about how this would appear if there was a deletion. Again, it's not going to be the whole chromosome that's deleted, just a part of it. So let's show our chromosome that contains a deletion. So I have our A and B, our C, our D, but let's say we have lost E, F, and G. We just have H here. When the deletion occurs in such a way that you've lost a middle part of the chromosome, that's called an interstitial deletion. We could compare that to if we lost the end. So if we lost A and B, that would have been called a terminal deletion because it lost the end, the terminal portion of the chromosome. But we're going to focus on interstitial deletions. Usually duplications or deletions occur on just one of the chromosomes in the pair. So if we looked at our duplication here, usually the other chromosome would look like this. Now, whether or not this is going to have a phenotype, we'll talk about later. But as you're looking at this, you might wonder what happens during meiosis. Remember, during meiosis, these two chromosomes have to pair up with each other during prophase. They have to synapse with each other. How can they synapse with each other if, as they align, E and F here, they align nicely with each other, but as it's drawn here, G isn't aligning with G, it's aligning with E. Same thing with H. It's aligning with F. So how is it going to happen that during meiosis, during prophase, when these chromosomes have to line up, and maybe or maybe not crossing over can occur, but they still have to line up regardless? How is that going to be possible? Well, the chromosomes do an interesting trick, and they will form a loop where this E and F will loop out. So let me draw what happens during meiosis when there is a duplication like this. You'll have your chromosome, like so, where you have your A, your B, your C, your D, your E, and your F. Now remember, this other E and F, we have to do something with that. And what happens is it loops out like so, where you have your E and your F. And then you have your G and H. Having this part of the chromosome loop out allows this homologous chromosome to pair up with it. So this other homologous chromosome now, as I just said, can align nicely with it. A through D, E, and F. And remember, this other one's not duplicated, so it just has G and H. There is no problem at all during meiosis. This does not create a problem. It's just a solution to the problem that it had. So meiosis will proceed as normal. Now, if it was homozygous and there was a duplication on both, then you wouldn't form this duplication loop here. The duplicated pairs would just align with each other. But when they're heterozygous, as they often are, you have to form this loop. Now let's think about deletions and how they're going to accomplish meiosis. They have the same problem and they're going to have the same solution. So this is our chromosome here, our deleted one, where we have our A, B, C, D, and H. However, usually deletions are on just one of the two chromosomes, but the other chromosome doesn't contain a deletion. When I say the other chromosome, I mean the homologous chromosome. And so what happens here is it's going to also form a loop so that the deleted genes E, F, and G are in this loop on the undeleted homologous chromosome. Again, this is just a solution so that proper synapses can occur during prophase 1. Meiosis will continue unaffected. This won't have any consequences to the meiotic products. It's just the solution so that synapses can occur. So the next thing I'm going to talk about is how do we form deletions and duplications. There are a few ways that duplications and deletions can form, but I want to talk about a way that they form during meiosis. And so I want to draw homologous chromosomes, much like I had on the last board, but I'm going to draw them showing all strands of the DNA. Just to remind you from our meiosis discussions, these two strands here are sister chromatids, and together these chromosomes and these chromosomes are homologs. We know that within a chromosome, crossing over occurs. And when crossing over occurs, this part of the homologous chromosome will swap places with this other homologous chromosome. Now on the last slide, we talked about the importance of them lining up properly. And that's why we formed those loops. One of the reasons it's really important that they line up properly is because if we have our genes here, we'll just draw a few here, A, and then we'll draw B, C, and D. As long as they synapse properly, and you have this crossing over effect, there should be no real problem. Because the products are going to be, in fact, why don't we just draw them here? Remember, during meiosis, this chromosome gets put into a gamete, this chromosome gets put in a gamete, this one does, and this one. This first one up here, A, B, and C, D, will get packaged into this gamete. This bottom one also will. Now these two middle ones that experience crossing over, they will also get packaged up into gametes, and because they line properly, A's to A's, B's to B's, C's to C's, D's to D's, that crossing over event in this case has no effect. Each of these chromosomes will be genotypically A, B, C, D. So how does this explain how we're going to get duplications and deletions? Well, we're going to have to reach back into our memory and remember something called repetitive sequences. So let's think about these repetitive sequences, and I'm going to put them right here. The thing about these repetitive sequences is that they share a high degree of sequence similarity. Let's make a key here. That's equal to repetitive sequences. All these repetitive sequences, as I just said, are by sequence very similar. Usually they synapse just like this, as they should. Every now and then what happens, because of the sequence similarity between this repetitive sequence and this repetitive sequence, things get shifted. And let me draw what happens when they misalign. They align with the wrong repetitive sequence. Okay, so when they misalign, let's draw what they look like. We still have our genes A and B and C and D, and it still has the repetitive sequences. But the problem now comes, this repetitive sequence between genes B and C now have misaligned with the repetitive sequences between C and D. And again, you still have this one here too. Now this doesn't happen a lot because you have to, as you look here, you can see there's not a lot of real good alignment with the other regions of the chromosomes. So it doesn't happen often, but it can happen. Now, at this area of the similar repetitive sequences, if we get a crossing over event right here, let's think about what's going to happen. It's kind of complicated, so let's follow this through here. And we'll start with the easy ones first. This top chromosome of the homologous pairs here isn't involved in the crossing over event, and this bottom one here is also not involved in the crossing over event. So they're going to have just what they should have, A, B, C, and D. Now let's follow this one here. It has A, B, C. It crosses over, so there's going to be exchange now where this part of the chromosome is going to get swapped up here. So again, we go A, B, C. In fact, let's write that first part. A, B, C. It then crosses down here and picks up C and D and moves it up here. It'll swap. This one will move down here. Okay, but for now, it's moving the C and D genes up to this top one. And as a result, as you can see here, we have 2 C's. It duplicated this region. Now, I drew this fairly simplistically with only 4 genes here, but you can imagine when this happens, you may duplicate more than one gene. You may duplicate a whole region. But in this more straightforward model here, we only duplicated 1 gene here. Now, let's look what happens here. We move along here, we have big A, big B, big A, big B. It crosses up here, and remember, we just swapped with that, so now crosses up here and picks up D. And so you can see what's happened is that we lost this region where the gene C was. So this gamete here is going to generate a duplication, whereas this gamete here will generate a deletion. These two will be unaffected. Now when they go on to fertilize with another gamete, the other gamete is more than likely not to have experienced the same kind of abnormal crossing over event, so the result should be a heterozygote for the duplication. Same thing here, should be a heterozygote for the deletion. Now, whether or not a duplication or a deletion has a phenotype depends a lot on which genes or which chromosomes have experienced this duplication or deletion. So what I'd like to do next is talk about the phenotypic effects that some duplications can cause and the effects some deletions can cause. Okay, so I want to focus first on duplications. And I'm going to talk about 2 things with duplications. The first will be a phenotype caused by duplications due to something we'll call gene dosage. Then I want to talk about duplications and how duplications are important in evolution. But let's talk about gene dosage first. Okay, so in talking about this gene dosage phenotype, let's draw a simple chromosome here where we just have 3 genes, A, B, and C. Gene A makes a protein, gene B makes a protein through transcription and then translation, and then finally gene C makes a protein. These proteins, A, B, and C, they come together. These 3 proteins now will come together and form a complex at the quaternary level, if you can remember that term from way back when. So it forms this multi-subunit protein. And one of the first times they noticed this effect of a duplication was in zebrafish. And under this circumstances, when you make equal amounts of A, B, and C, the zebrafish develops normally. Now, if by chance there was a duplication and you have an extra copy, say of gene C now, what happens is you produce more protein of gene C because you have 2 copies of gene C. And now what happens is that other copy now will either try to interact with this one or maybe it prevents it from forming at all. But the end result now is that it doesn't develop normally. Instead, there are several developmental consequences. Some of them are simple, like the pigmentation changes, but there are other consequences as well, all because of one extra gene being produced. It's important to remember, and we'll touch on this later, and if you take a developmental biology class, which I highly recommend, you'll see this even in greater detail. That development from that single zygote to a fully formed organism is a critically timed and critically dosed event. Changing it even a little bit can have great consequences. There are many human diseases associated with duplicating just one region. Sometimes it's more than one gene, but sometimes it's just a single gene or 2. That throws off development and can lead to fairly severe consequences. Next, I want to talk about duplications and evolution. So let's talk about evolution. And in doing so, I want to talk about Onos Hypothesis. So in Onos Hypothesis, let's consider a single gene. And we can call this gene just generically gene A. It seems to be our favorite gene name. So we'll keep going with that. And let's say in this instance that gene A is essential, meaning if it's mutated to the point where it's non-functional, it leads to death of the organism. Mutating an essential gene will lead to death. From an evolutionary viewpoint, it's hard to generate mutations in this gene to see if you can get an improved function. That's where duplications come into play. So in duplications, let's make a tandem duplication. It wouldn't have to be tandem, but it's just easier to draw it that way. We now have two of these essential genes. So the way this works is you duplicate an essential gene. And it's also important that there is no dosage effect. Some you can duplicate and there's no effect. It just depends on the gene. Alright, so now both of these genes are still essential. You still need gene A. However, now if we have an extra copy of gene A, what can happen is on this extra copy here, it can gain mutations. Maybe one, maybe several. Now, over time, what can happen? And when I say time, I don't mean one generation or two generations. I'm talking about many generations, so thousands of years. What can happen is, at some point, this gene A here has gained what we might consider to be advantageous mutations. And while this gene A here is still doing its job, so it's still doing its normal job, this gene here has gained these mutations. And now maybe this mutated extra copy, duplicated copy of gene A, which we'll maybe call A1 now because it's different now, maybe now it has an improved function. Maybe it works better than A, or maybe as multicellular organisms evolved, maybe gene A1 now works better in a different organ or a different tissue, or maybe at a different developmental stage of the organism. Regardless, what I'm saying is this improved function leads to a reproductive advantage. For whatever reason, this improved function makes this organism more fit, meaning, when I say more fit, I don't necessarily mean stronger. I mean that it can produce more offspring. And by producing more offspring, it passes this newly improved gene on. And so that organisms that have this new gene continue to be more fit than the others, and they continue to pass those genes on. In class, we will work on some activities that talk about some real examples in human evolution where this has happened. But what I want you to know now is that Onos Hypothesis explains new sources of genes. Okay, now I want to talk about deletions and how deletions can lead to some sort of a phenotype. So we're done talking about duplications. Let's focus on deletions. And so as we think about phenotypes associated with deletions, it's important to consider types of deletions. The first one I want to throw at you here is called a centromeric deletion. So for instance, if we have our chromosome here with our centromere right there, and the deletion results in regions of the DNA or chromosome being removed that include the centromere, like that would be called a centromeric deletion. Centromeric deletions, because they've lost the centromere, ultimately the whole chromosome will be lost because it won't segregate properly during meiosis or mitosis. So as a result, centromeric deletions are usually lethal. The next type of deletion I want to focus on are homozygous deletions. Now, as I suggested before, homozygous deletions are rare. They occur rarely, but another reason they're rare is because they're usually lethal. And the reason for that is if you have a chromosome shown here, and let's say we delete this section here. And usually deletions aren't just removing one or two genes, it's usually removing a whole series of genes. And so if you have both of your copies here, of your two chromosomes, and you delete both these regions, the result is a much truncated chromosome. By itself, that's not a problem. But because you've deleted both sources of genes on both of these chromosomes, there's a great probability that some of those genes are going to be essential. And now you don't have any of them. So because of that, they are usually lethal. Okay, next let's talk about our third type. And these are heterozygous. These are not necessarily lethal. They could be, or they can just generate other kinds of phenotypes. So I want to spend a little bit of time talking about heterozygous deletions. But first, let's remind ourselves what that might look like. So you might have a heterozygous deletion here where this chromosome has all of the genes necessary, and then on the other chromosome, the other homolog, it's missing these genes. There are three effects associated with heterozygous deletions I want to talk about. They are pseudodominance, an imbalance of gene product, and haploinsufficiency. Let's start by talking about pseudodominance. And to do that, let's draw some genes here. Okay, so now you can see the placement of the genes on these two chromosomes. And for this scenario, let's just say all of these genes, A through E, are recessive genes. Meaning, as long as you have one dominant allele, big A, the other allele could be little a, it could be absent. The key is you have one copy of the dominant allele. And in that case, the individual will be okay. However, if an individual has two little a's, the two recessive alleles, then they will have the trait, the phenotype that's associated with not having a functional A. This particular individual, for A, B, and C, they have two dominant copies. For instance, they're big A, big A. Over a course of a lifetime, they may get a mutation in A, and it becomes little a. So they may become this. But phenotypically, these two individuals are okay, because they have one dominant allele. Now, in order for them to get the recessive trait, they would require two mutations. Remember, the first is going from big A to big A, little a, and now the second mutation goes from little a to little a. It has to happen in the exact same cell as well, and that's pretty rare. You wouldn't expect that to occur frequently. However, as we look at these deleted chromosomes, we're missing D and E here. So this individual is big D, and then we'll just put an X. They have nothing there. They just have one of the alleles, big E and an X. For them to express the phenotype, they only have to get a mutation in D. And now, little d and nothing, because it's not there. They will have the phenotype now. The trait. Again, in each of these cases, they only, over a course of a lifetime, have to have one mutation in this big D or one mutation in this big E to get the associated phenotype or trait. We call this pseudo-dominance. It's much more likely to get one mutation in one allele in the same cell versus getting two mutations in the same cell. So this pseudo-dominance effect increases a person's chance of displaying a recessive phenotype. So next, let's talk about imbalance of gene products. Let's say, for instance, that gene D makes this protein. And for the sake of this scenario, let's say that gene D interacts with gene A, gene B. And in order to be functional, you need two copies of each. And so they have to be in the same proportions. But since we have one less copy of gene D in the chromosome, what will happen is for every two Bs that are made, protein Bs that are made, you only make one D. And to be functional in this scenario I just drew here, you need to have both copies of D. So you have this imbalance of gene products in our mutants here. And so this will lead to some sort of a phenotype. Whatever that phenotype might be depends upon the function of this original protein complex. Again, this imbalance of gene products creates a protein structure, a multi-subunit protein structure that doesn't contain enough subunits. Next, let's talk about haploinsufficiency. The best example of haploinsufficiency is with fruit flies. Okay, so let's draw a fruit fly. And you will no doubt be super jealous of my fruit fly. And I'm going to draw its wing here. And usually a fruit fly's wing will have a very regular structure to it. And in this example here, let's say that this forms properly because of a gene called Notch. And as long as Notch is present, it forms these receptors on the outside of the appropriate cells that are involved in wing development. And when it's time to form wings, having these appropriately numbered receptors allow signals to enter the cell to signal proper wing development. Now the reason you get the appropriate number of receptors is because we have two copies of Notch. If there's a deletion and we only have one copy of the Notch gene, what happens is in your cells that need to respond to make the wing, there are half as many receptors. And so when it receives the signal to make wings, there's not as many receptors. There is an insufficient amount of receptors. That's how it gets its name. Insufficient amount, compared to 4, as I drew here. And as a result, the fly will have an abnormally shaped wing. It looks kind of notched. That's how it got its name, notch. Haploinsufficiency, because the deletion causes less proteins to be made, and so in the end, less receptors are made, an insufficient amount of receptors. So it can't respond to the signal in the same way that the unaffected fruit fly can. So you get an abnormally shaped wing. Okay, that's all I really want to say about duplications and deletion. If you have any questions over this material, please make sure you come see me. If not, I'll see you in class. Bye.